So a flow cytometer is a machine where we take a blood sample, we clean it up to isolate the lymphocytes, then we inject the lymphocyte in that machine. All the cells line up into a very, very small tube. There's a laser that goes in front of that machine, and then you measure the shadow of that cell for its size, its luminosity, because all the different immune cells have a different type of luminosity, and you can use antibodies for different stem cell markers or non-stem cell marker, and through this process of inclusion-exclusion markers, then you can identify which one is a stem cell. So you prepare your solution by incubating them with these markers, and then the machine will basically just aspire the sample, pass it in front of the machine, and then the machine will count all the cells that identify as a stem cell. The patient comes in the morning for an hour, they take a blood sample, we give them placebo or the plant that we're testing, and then we take another blood sample an hour, 2 hours, 3 hours after. We count the number of stem cells, they come back a week later, we do the exact same thing to establish a circadian cycle, and then we subtract that circadian cycle from the response that we get with the plant. So it tells us how many more stem cells we have in the blood circulation when on the day that the people took the plant.